Welcome, and thank you for joining us for today's presentation. My name is Elizabeth Firth. I'm the Senior Product Manager of Human Genetics at DNA Genotek. DNA Genotek is the leading provider of products for biological sample collection, stabilization, and preparation. Today, you're going to hear from Professor Antonio Belli about his work in small, non-coding RNA biomarker discovery. The title of his talk is Saliva RNA for the Detection and Management of Concussion: Discovery, Validation, and Commercialization. Before we begin, a quick housekeeping note that you can submit your questions via the question box throughout the presentation, and questions will be answered by Professor Belli at the end. A recording will also be made available through ESHG's on-demand webinar resources. Now, without further ado, I'll turn the floor over to Professor Belli. He's a National Institutes for Health Research Senior Investigator, Professor of Trauma Neurosurgery at the University of Birmingham, Consultant Neurosurgeon at UHB, and Director of NIHR Surgical Reconstruction and Microbiology Research Center. He's an advisor and guidance author on concussion to England's Premier League Rugby Football Union, Football Association, and Cricket Board.
Many thanks for the invitation to speak today. It's a great honor and privilege to be here. And thank you for inviting me to talk about our research, which obviously always makes me happy and enthusiastic. Our research really focuses on the discovery of traumatic brain injury biomarkers in biofluids, but particularly this talk will focus on the work we've done on saliva and salivary biomarkers in the context of sport concussion, which hopefully will have application for many other fields. Let's start with some disclosures. I'm a professor of trauma neurosurgery at the University of Birmingham and work at the Queen Elizabeth Hospital, Birmingham, which is a major trauma center. I'm a practicing neurosurgeon. I'm the founder and director of a company called Marker Diagnostics. I have received honoraria for various talks by companies, and these are the main ones are listed there, and various grants from different sources. I don't have any financial conflict of interest with sports authorities and no financial involvement with DNA Genotek. So traumatic brain injury is indeed an increasing pathology. We are seeing traumatic brain injury numbers going up and up over the years, and is now projected to have become the largest cause of death and disability by last year, really 2020. And it's certainly the leading cause of death under the age of 45. There is also increasing concern around concussion in sport, and about 10% of all traumatic brain injury cases are now attributable to sport concussion. What is actually alarming that the rate of concussion seems to be going up relentlessly year on year. This is due to a number of reasons, including sports becoming more physical. There's more emphasis on physical preparations. Players are getting faster, they're getting bigger, they're getting heavier, and also possibly more awareness of concussion, which is actually itself a very good thing. Some of this may have been driven by litigation, both in the United States, and now we're seeing that in Europe as well, and in the UK in particular. But despite that, there's still some concern that concussion is missed. And I just want to give you a very quick example of what might happen. This is from a Champions League semi-final a couple of years ago. Obviously, an event is probably watched by a billion people rather. And this is Vertonghen, who suffered a concussion during the match, but he was assessed and was thought to have a sort of a maxillofacial injury, only was allowed to return to play. But as you can see, just a few minutes later, it has to be helped off the pitch. And initially, concussion was denied, but then Vertonghen admitted that he did suffer a concussion, and in fact, he struggled with symptoms for many, many months, and he probably still does. So there are several concerns in regard to sport concussion. Clearly, early damage and a big bleed is what many doctors on the sideline are concerned about. But catastrophic injuries are relatively uncommon and fortunately very rare in professional sports. But certainly, there are implications of missing a concussion and letting a player continue with the concussion, and it has implication for prolonged recovery, more severe symptoms, potentially even severe consequences. For example, a second impact syndrome, which happens when a player is concussed more than once in quick succession, which could potentially lead to a catastrophic outcome, such as the case of Ben Robinson, a rugby player who was concussed 3 times during a match and unfortunately and very sadly didn't wake up after the third event. And then there's the whole kind of worms on neurodegeneration, which is what many players and parents are concerned about right now. The difficulty is that there is no reliable test of concussion on the market yet. There is not a single, it's a single event, but not a single pathology. Concussion is a basket term for many different presentations and clinical manifestations that can take very different forms, and no big concussions are the same. Concussion can be very subtle and easy to miss. It also evolves over hours or days, so a player can actually carry on sometimes during a match if the concussion is not diagnosed, and then only to develop symptoms in the evening or the following day, having exposed the player to the risk of having potentially suffering another concussion during the same match. And this is an example, even in professional sport is well organized, such as rugby. It is, and with video replays and cameras watching the player from every angle, it is possible still to miss a concussion. This is probably one of the most rugby players, called George North. You can see a small dent on the pitch there where he lands. He was assessed immediately after the concussion, was passed all the tests successfully, and was allowed to return. But obviously, looking at the video footage, you can see that he loses posture for a few seconds, which is one of the classical signs of concussions. And obviously, the dent on the pitch probably leaves no room for ambiguity there. But this is just to say how a concussion can be missed, even with the best technology available and the best possible training you can get in a professional setting. We still really don't know what happens after concussion to some extent. We know that on a small level, it's very much an electrochemical event. As you strain an axon and stretch it in different directions, you'll get an electrochemical discharge and a release of neurotransmitters, and that can cause an impairment of consciousness, a bit of sort of transient amnesia, lots of posture, sometimes even convulsions for a few seconds. But actually what we're worried about is the damage that occurs on the inside, the straining of microtubules, for example, and the consequences that this carry for axonal transports in particular. So a cell will actually experience a sort of functional damage rather than a structural damage that is visible from the outside. This is an example of what happens in an axon with traffic of mitochondria and vesicles, but after injury, everything grinds to a halt. So the cells look still intact on the outside, but metabolically they are dysfunctional at this point. And also the recruitment of neuroinflammatory cells and microglial cells to the site of injury that increase neuroinflammation, which can potentially have long-term consequences. And this is just an example of that. You know, this is a PET study done on people that had a mild TBI, and even years, in fact, even 16 years after injury, you can see signs of neuroinflammatory ligands being present in the brain of patients in certain regions. So obviously this inflammatory state can be persistent even in a dormant state for many, many years. And as I said, there is the big issue of sport concussion. We know from epidemiological evidence that there is an accumulation of cases now of retired American football players and soccer players and players from other sports that develop this condition called chronic traumatic encephalopathy, which is actually a tauopathy. There's animal evidence as well of this tauopathy being transmissible between animals. If you take brain from mice they've been concussed repeatedly, they can actually give tauopathy to recipient mice. But it's also a counter-argument to that, the fact that the evidence is still limited. There is selection bias in many of these clinical studies, and other factors can explain the pathology, including lifestyle, comorbid factors, drugs, or weight gain after retirement. And obviously, the benefit of sports always need to be considered, and these are obviously extremely important in this decision making. In fact, one of the best reasons to prevent dementia is to remain active into a late life. However, recognizing concussion is extremely important, and herein lies the problem. So if we're not able to recognize concussion properly and diagnose concussion properly, the rest of the management strategy really falls down. And this is from the sort of concussion consensus meeting, which really stipulates that the first two steps are to recognize when a player is concussed and remove the player immediately without allowing the player to return to competition on the same day. And then the rest follows, but it's obviously if you're going to miss a concussion, you're not going to be able to implement any of that, and the player will miss out on targeted treatment as well. The symptoms of concussion are non-specific and can be confused with a number of things, including match stress, lack of sleep, dehydration, fatigue from the match, intense exertion, and so forth. And unfortunately, many of the tests that are currently used in clinical practice, including rugby, have limited sensitivity or specificity, or both in many cases. So there's still need to corroborate the current assessment with something objective, something quick, something reliable, and something accurate. And even more so in the non-professional setting, because when you drop down a couple of leagues, the performance of this test actually drops significantly in the hands of non-professionals, for example. There's been much attention in recent years on traumatic brain injury biomarkers, and there are a number of compounds that have been studied that are derived from cell damage, either the cell body, the axon of a neuron, or the astroglia, or even oligodendrocytes. And we've been looking for these compounds in the blood for many years, and they all have variable accuracy and variable sensitivity and specificity. But the main problem is that many of these are actually not very specific. So they also produced by other organs, and particularly in the context of sports injury, they can come from a number of different sources other than brain. So for the last few years, we've become interested in different type of biomarkers, particularly RNA biomarkers, and specifically initially we started with macRNAs, which, as you all know, these are sort of intercellular messengers that are there to modify the response to injury, cancer, or stress, or other conditions by effectively switching the action of genes off and effectively modifying the response to that event post-transcriptionally. So there's been increasing interest in macRNAs in particular, and this is just probably illustrates it very well. MicroRNAs are produced and packaged within the mother cell and then sent out in exosomes or other lipid bodies. And once they reach the target cells, they get internalized and they exert their function in the target tissue. And there's sample literature now in particularly on blood and microRNAs, because it's obviously a convenient sample that it's easy to get, for a number of different conditions that include inflammation, oncology, and now so there's also increasing literature on trauma as well. So a few years ago, we started off with a study of traumatic brain injury in a healthcare setting. We sent paramedics out to get a blood sample from road traffic accidents, and they were actually on scene within the very short time. Within just a few minutes, they were able to get us a sample. And what we saw there actually, yes, there were indeed microRNAs that were very exciting for us, and they were very accurate in areas under the curve, close to 100%, very fast, and they were indeed, telling us a story that multi-BI could be differentiated from severe TBI or from extracranial trauma or from healthy controls, for example. The problem with this is actually that blood is not a very good fluid to use in a sports setting or many other settings. It just, unless you have a phlebotomist on site or a doctor on site, which obviously community sports would not have, this is probably not the first biofluid of choice for many of these settings. So we then became interested in saliva, and then the reason for that is saliva contains a lot of macroRNAs and other small non-coding RNAs. In fact, more than most other biofluids and tissues in the body. It has many functions. We produce 1 or 2 liters of saliva every day. And it's not just used as a lubricant, it's antibacterial, it has signaling function to the digestive tract, it regulates the gut microbiome, and it probably has many other functions that we still don't know much about. So we started looking at saliva, and initially we started looking at patients that came to the clinic with a traumatic brain injury from a sports setting. And we could see that actually there were quite a few macroRNAs that we became interested in. And these were decent, sort of differentiating these cases of traumatic brain injury from healthy controls. But obviously these are sort of 2 to 3 days down the line. So we decided to partner up with the Rugby Football Union in England and launch a study looking at these macroRNAs and small non-coding RNAs in the context of professional sports. And the reason for that is that rugby provides an excellent setup for this kind of study, because some players are assessed immediately, they're assessed at different time points after the injury, and they're evaluated in a very professional and thorough way. So really, in terms of having a comparator, this probably offered a very good opportunity for us. So we collected samples across 2 seasons, and we evaluated the concussions against other control groups. For example, for players that have been removed from the game due to musculoskeletal injuries, or players that played the match but not suffered any particular event, but have been exposed to match stress, dehydration, lack of sleep, and intense exertion, and so forth. And we were looking in particular at players that had taken the HIA test, so the head injury assessment test, and whether these biomarkers could differentiate the players that passed the test from the ones that had failed and therefore were deemed to be concussed. So initially, not knowing exactly what we were looking for and not wanting to rely on past experience, we did next generation sequencing and then ended up with a number of plausible readouts after controlling for multiple comparisons. And then we gradually narrowed down the list, looking at for example, what was expressed after concussion versus what was present at baseline. Every player had a baseline at the beginning of the season. So excluding, for example, biomarkers is simply, we couldn't rely on because they were already expressed in many players pre-season, or they were present in musculoskeletal injury. And so it was a process of elimination, which left us still with a long list of biomarkers. And then we used these biomarkers to generate an algorithm that could predict which players were actually concussed versus all the control groups. And this reduced the list further to about 14 biomarkers that eventually they seem to be critical or at least very useful at making that diagnosis robust. So obviously, there wasn't really one biomarker that on its own could actually tell us the full story, but it was a combination the algorithmic combination of different biomarkers, because these small non-coding RNAs act in concert, so they need to obviously affect a response to the injury by switching sort of a multitude of genes off or on after trauma. So they actually, one biomarker may not give you all the answers you need, but actually, if you look at how they move in concert, actually you get a much clearer picture. And when you do that, you see that combining these biomarkers this way will indeed tell the concussed players apart from the other groups, all the control groups and the players that were negative on the HIA. And this was with a high degree of accuracy, 100%, immediately after the concussion, but also at time point 2, which is immediately after the match, and time point 3, which is 36 to 48 hours after the match. And so we worked this out in the first season, and then in the second season we sought to validate this prospectively and blindly on the next group of players. And actually, in season 2, we were very pleased to see that the accuracy of the biomarkers really held up, and it was above 90%. In fact, it was at time point 2, it was 96%, which obviously is something we were very pleased about. Clearly, there is a value of this at elite level, but also at community level, probably even more, where actually there are currently no professional assessments are available to youth player or academy players or even Sunday league players. And also we think there may be opportunities to look at this in the context of predicting recovery and predicting the effects of multiple injuries in a long-term brain health. This is work in progress, of course. So from a commercial point of view, what we thought we could try to develop a method that uses is derived from actually the DNA genotek mass swab that we actually use for the study, which allows the saliva to remain stable in terms of snCRNAs for several weeks, and it's actually very easy to use in a clinical context because everybody's now getting used to doing mass swabs, and it only takes a few seconds. So how we think this is going to work effectively is that a player would collect their sample, send it to a lab, and then it would get the results fed back to them or to the doctor within a short time. So the research has had a lot of publicity, and we're very fortunate that a lot of media outlets were interested in our research, and this went viral in many continents. So we were very pleased about that. But clearly, there is still work to do. For example, we still don't know how the salivary biomarkers get there. We think they're actually generated in situ from salivary glands, and why they get expressed in such large quantities, we don't know. But if you accept the findings of our golden hour study, for example, we know these biomarkers, snCRNAs are present in blood immediately, and therefore, if these snCRNAs target a number of tissues downstream, including salivary glands, it is possible that triggers a response in salivary glands, just like it would do in any other tissue. There's also a very large parasympathetic innervation of the salivary glands, and we know the vagus nerve is implicated in the response to brain trauma in many different ways. So the connection to the salivary glands, from a physiological point of view, makes sense, although there's clearly more work to be done in that respect. We also don't know what these biomarkers, what these snCRNAs exactly do. The number of pathways that we could explore are involved in neuronal repair, neurite arc growth, and axonal guidance, but also interestingly, other mechanisms. For example, there seems to be a switch to fatty acid metabolism after this kind of injury, which it's interesting, and it might be the object of much more discussion than we have time for, but probably would make sense from an evolutionary point of view after trauma. But there's more work to be done on discovering the actual targets of these snCRNAs. We are now in the next phase of work where we're actually collecting samples across many different sports, including football and rugby, boxing, and other disciplines. And we clearly want to expand our sample to females, because the Scrum study was only all males. We did attempt to do more work on females, but it didn't quite work out because of the limitations in the number of samples we were getting. But clearly, we want to expand this work to youth players and female athletes in the next wave. So this is all I have time for, but it's been an absolute pleasure to speak to you today, and I would really like to thank DNA Genotek again for inviting me to speak today. And a number of co-workers, and you see, it's a long list there. But I'm very grateful to the players, the clubs that helped with this research, and the funders and all the co-workers, and obviously the academic partners as well. Thank you very much.
Quite amazing to me how quickly these biomarkers appear in saliva. You know, the fact that it's within minutes is really quite amazing, and how accurately they can be detected as well. So I think at this point, we'll move along to questions for Professor Belli. So just a reminder, if you could please submit your questions via the questions box in your web browser.
While we wait for any questions, I think that's, I mean, that to me is quite remarkable as well. I mean, how quickly you can detect these biomarkers in saliva, I think is probably, obviously, you know, salivary SNC RNAs have been discovered in a number of conditions, including concussions. We published some work before, other people have done the same, but what really surprised us actually how quickly these could really appear in saliva, and the mechanism for that is obviously a bit of a mystery, but it's just absolutely fascinating that, you know, within literally, by the time the player walks from the pitch to the medical room, those changes are already there and they're measurable.
Yeah, that's quite astounding, actually, that it happens that quickly. One of the questions that I had for you was, you know, the microRNA profiles that you're looking at. Is there any ability to predict from that the area of the brain that sustained an injury?
It's a very good question, and this is we can't quite look at the area of the brain that suffered an injury, and that's probably a limitation of you know, of neuroimaging in a way, so that we can't really make those correlations pathoanatomically, because the neuroimaging, you know, even advanced MRI scanning, which is what we do, doesn't really give you that kind of level of resolution as to what area has been damaged. But this is it would be fascinating to trying to make the correlation. And I know that I'm aware of some imminent publications by other groups, for example, that may have worked out a way to, you know, use neuroimaging to actually work out which part of the brain is mostly affected by concussion. Then we can probably use the biomarkers that we know of to trying to link the two. But so this is actually a really good question, because hopefully, you know, sort of the way this is going, we might be able to make that correlation at some point in the near future.
Okay, and I think you had mentioned in one of your slides that there may be some differences in this that you can detect that indicate the severity of the concussion. Is that true? Like, there, you know, depending on the profile you see, it's different between a mild injury and a very severe one?
Well, certainly, for the Scrum study in rugby, you know, the level of we looked at players, for example, that had, you know, for example, that were an obvious concussion with loss of consciousness, for example, and the ones that actually were only removed after medical assessment, thinking, well, those must be milder. So there were some differences there, but they were not very, very clear. From the other study that I mentioned, which is the Golden Hour study, there were some very clear differences between mild and severe. So some microRNAs were in common between the two, but a lot of them were different, which may surprise a lot of people. But actually, that probably is a signal that microRNAs and snCRNAs really describe the body's response to injury, rather than, you know, biomarkers are released from tissue damage. So in a way, the body will respond differently to a severe neurological event than a mild one. You know, clearly, you know, if you remain conscious and whatever. So I think it's what these microRNAs are telling us, that the genes that get activated or switched off after a mild event actually need to be different from the ones that, you know, that perhaps need to operate when a patient's in a coma, for example. So I think it's probably the way I would look at it. So there are clear differences there, but in the Scrum study, they were all towards, there's a ceiling effect. They were all quite mild.
Okay. And we do have one question here from Grant Mitchell. Is the change in microRNA specific to brain injury? Do you see similar changes with fractures, visceral injury, or with epilepsy?
Yeah, that's another very good question. So in our study, we used different control groups, and one of the control groups was players with musculoskeletal injuries that have been removed from the match due to that. So the ones that were ultimately, there were quite a few that overlapped. So it didn't matter what type of trauma you had. So we excluded those from our analysis, from the algorithm. So the ones that we actually ended up using are the ones that are specific for the brain, and they're not affected by other injuries, because this is one of the things that actually affects much of the discovery of biomarkers in this field. You get excited about something, but then you discover that similar biomarkers are produced by orthopedic injuries, for example. So in our process, we use a control group that could have, one of the control groups was actually orthopedic injuries.
Okay, and I think that the time for our presentation is elapsed. So thank you, everyone. That concludes today's webinar. Thank you, Professor Belli, for sharing your expertise with us. I appreciate you joining us. And I'd like to remind everyone that a copy of this webinar will be available through ESHG's on-demand webinar resources and can also be found on DNA Genotek's Company Corner. Additionally, all attendees will receive an email with a recording following the session. Thank you, everyone, for joining us today.